we are now switching to classification because this becomes super, super important when it comes to COVID, because we need to work out the phylogeny. So we're looking, if you like, at the evolution of the coronavirus. So how do we do this? We do this with genome sequencing. So that's one thing, actually, you need to be aware of this. That's one thing where we have a huge advance from 2002, because 2002 genome sequencing was massively, massively expensive. And so we'd have been prattling around with protein matching. Instead, we're banging there straight away, sequencing the genome of coronavirus. Okay, so that's a huge improvement. So the early sequences suggested it might have come from a pangolin, which is this creature very on the right, it's found throughout Asia. But now we're pretty much 100% sure it's the Chinese horseshoe bat smiling at us here on the left. Okay, is the evolution, if you like, of the virus. So how are we doing that? We're doing that with sequencing all these CGs, A's and T's and looking for a match. Okay, and so there's a database full of all these animals and their genome sequence. And so we have a match, which is the Chinese horseshoe bat. So how did it get from the bat to humans? Sort of a rumor mill. So that is a wildlife market in China. So this is not a wet market. Actually, we conflate the two. Because a wet market is just, I mean, I've been to Hong Kong before, and you know, you go into a restaurant and it's the best fish ever because the fish is still alive in the tank and they kill it for you, cook it then and eat it. And I'm sorry, that fish tastes better than other fish in my opinion. I like it really fresh. So the idea of a wet market is, as opposed to a wildlife market, is there's a chicken there, they keep the chickens alive and you rock up and they kill the chicken on the spot for you and give it you. And you go and cook and eat it that day and you know it's fresh. And that's kind of important if you think how hot it gets in the summer and how quickly meat can turn bad, especially if there's no refrigeration. So wet markets in general are certainly in Hong Kong, they're not wildlife markets. They're chickens, pigs, etc. And the problem is, if you've got all these chickens and all these animals next to each other, they can spread viruses between them. Now, the wildlife market, some of it's for eating, some of it's for medicine. You see some really strange species there. And I remember, not in Hong Kong, but in Singapore, I've seen snakes on the menu, crocs on the menu. So that's one possible way the horseshoe bat mingle with humans. The other possibility is from a lab in Wuhan. Scientists don't think this was a bioweapon. And the reason they don't think it's a bioweapon is the genome sequence is so similar to the bat in question. Okay. And so the only way this could be a lab-made thing is if they took a bat and slightly adapted it. But we don't think, well, most people don't think that's happened, but there's plenty of interesting conspiracy theories on that. Okay. Genome sequencing to determine the what species it is. So we're looking at a genome for this virus of 30,000 nucleotides. It might seem like a lot, but obviously the human genome contains 3.2 billion. And here's the sequence of a protein spike, 3,500 bases is what we're looking at. So we have to match all of this with a bat. So it's obviously a job for computers. And so the key difference in SARS-CoV-2 that has allowed it to cross species from bat to human is 12 bases, folks. Here they are, okay? CCU, CG, well, I won't read them out. Because that difference now helps it bind to human cells, okay? Whereas previously, you would expect the virus a bat virus to only infect bats, and it would only infect bats because it would only bind to bat receptor cells, and bat receptor cells might be slightly different. So we think it's a mutation, folks. We think the virus has mutated, and the mutation that has occurred has allowed it to bind with humans when previously it was just present in bats and couldn't bind to humans. Okay, so it's transferred from one species to another. And obviously all of this code here codes for this shape that you get here, the protein spike. So here's a sort of family tree of the different strains. And so the principles here are the same as any other classification question. This is further away in time, and I'll show you in a minute, but it's pretty obvious that this purple strain, these purple strains are all in China, folks, where it originated. Okay, we can see, you know, in some strains down here have died out now because the disease is under control. Okay, some strains have got right to the end, to the present day, and are still raging right around the world. And so these different strains have all been worked out using something called a PCR test or genome sequencing. Okay, now, should we be alarmed at the number of different strains there are? Is it mutating? Oh yeah, yes, it is mutating. So you think, oh my God, sir, why is there so many different strains here? And is it getting worse? Well, you've got 30,000 bases, haven't you, to copy every time you make a new virus. And so every time there's a mutation in a change in the bases, that is just a different strain. Now, the only possible thing, or not the only, the main possible thing that could affect whether it got more infectious, which was the subject of the 2011 film Contagion. The virus got more contagious in the middle of the epidemic, got more deadly. The only thing that would affect that would be some kind of mutation in those bases that make the spike, and not only the spike, the binding site of the spike. So pretty much any other mutation that you get, unless it makes the virus more robust or something, or it's not really going to make it more infectious.
Pardon?
Now you can see the countries. So just a point I wanted to make about that. You can see that's just the USA had one source of infection from China, and they had, don't want to get political, but they had about a month where they did nothing, and they could have contained it better in that period, and now it's boomed. And America also has had this infection here from Europe. Okay, so you see Italy will be, so we've got China in purple, America in red, Europe in green, all slightly different strains, but as I said, it could just be some RNA letters somewhere else. It's not making it looks like there's a lot of difference, but this is a quite standard amount of difference, and it isn't something that we're too alarmed about. But you can trace, so through this sort of classification, if you like, you can trace the flow of the disease from China to the USA. The point I'm making is a hell of a lot of the New York deaths, which is loads, by the way, seem to have come from Europe. They didn't come from China at all, and so we can tell that. And obviously, most of the UK we're linking to Europe really rather than China from this. You've got UK in there somewhere in yellow. It's impossible to make it out. So back to this as well. I've already talked about that. So that's what's going to determine if the transmission goes up with different strains, which we've already talked about this, whether the different strains will affect how infectious it is, will all depend on this spike area. And so that's the only thing we'd need to worry about. At the same time, having a unique pattern there is probably going to be the target for a vaccine. So we hope, in a way, we hope that area doesn't mutate too much for two reasons. So it doesn't become more infectious, and also so we can get a vaccine on these protein spikes, folks. So on your course, you have ELISA, enzyme-linked immunosorbent assay. So this is for the antigen test and the antibody test. Now this is kind of out of date because the antigen test here they're saying it's direct ELISA. What they're doing with coronavirus is a RNA sample. So what they're looking at, they're looking through, if you go back, all of these bases to try and find a unique sequence of bases that will only be found in coronavirus, okay? And then they can search your human nose cells or blood cells to search if you've got the antigen, and how it will play out will be an RNA match. So this, I think this direct ELISA is already starting to be out of date a bit. This curriculum was written in 2016. So the indirect test is going to be really, really valuable, isn't it? So that's the test to see if you or I have antibodies for this disease in our bloodstream right now. Because if we do, we've already had the disease, and we're very unlikely to get it again, although people are casting doubt on that, which alarms me. I've not had a chance to read too much into that. But yes, in Milton Keynes and all around, they will be doing some kind of indirect ELISA antibody test, okay, to indicate whether you've had a previous infection, which is different to the direct ELISA or PCR RNA test actually for antigen, which is, have you got COVID in your blood right now? Okay, so two different tests. Remind you how this one works. There are other variations of this theme. The antigen is placed on there. You put your blood into it. Okay, if you have the antibodies, they will bind to the antigen there, and then you'll have another antibody carrying an enzyme that forces a color change that will bind to these antibodies. If we get a color change, it means we do have the antibody. Now, if we didn't have the antibody, you would wash it out to get all of the antibodies that are not bound, have to come out of that. They have to come out of that there. If they haven't bound to the antigen, you've got to wash them away. Otherwise, you'll get a false positive there. Okay, and so that was the indirect, in a nutshell, the indirect ELISA test, which is the antibody test. Okay.